Thank you, David. And that's the first time I've had someone pronounce my name correctly. McPherson. Okay. So I'm going to start off with a little bit of a review on GWAS. Some of you have heard this before. And for this sort of mixed audience, I'm trying to come up with a few pearls in terms of what we've actually learned over the last 10 years, 11 years, and perhaps how we can use these data. So coronary artery disease, which is one of the two complex phenotypes that my laboratory is interested in, the other is obesity. You know, these are complex diseases, and unfortunately rarely due to a single mutation. I suppose there's a few exceptions. Familial hypercholesterolemia is autosomal dominant, monogenic, and of course patients with FH not treated often have quite early coronary disease. But for the most part, we're talking about a disease where there are multiple alleles, pretty uncommon, certainly less than 0.05%, and most are less common. Some are less common than that, some are a bit more common, but they each have a small effect. So this makes it difficult to actually sort through a lot of data to find the variants that are important. So what we start with, genome-wide association studies. As you know, we started this at the Heart Institute way back in, actually it started in about 2002. And the first genotyping we did was with Perligen Bioscience in San Francisco, and we had a chip that had 70,000 SNPs on it, but it was enough to find things that were very interesting, particularly 9p21. So these SNPs, of course, these chips now have about 1 million SNPs. They cross the genome, but of course those individual SNPs that are genotyped are not necessarily causative SNPs. So really we're just kind of getting a map of the genome, and the genotype SNPs shown here, the SNPs on the array, may not be functional, but often they're in linkage disequilibrium with a functional SNP. So what we do in the lab is actually trying to find the functional variant and then trying to figure out exactly how it exerts its effect. For the most part, for a complex disease like coronary artery disease, the variants that affect risk and have a small effect are variants that affect gene expression, RNA processing, etc. They're really things that have a functional effect at the level of molecular biology rather than a mutation in a single gene. And so we actually end up with about 15 million SNPs that are generally pretty good quality. And how do we do this? It's because there are haplotypes across the genome, and we have information on that. So we phase to the estimate the haplotypes, and then with the 1000G data, so we have whole genome data from the 1000G project. We'll soon have it from the UK Biobank, actually at some point in 500,000 individuals, but we can phase the haplotypes and link these to 1000G data, and basically we end up in our studies 9 to 15 million SNPs with a minor allele frequency that is greater than 0.005. So this doesn't allow us to interrogate very rare SNPs, and of course, for that, exome sequencing, whole genome sequencing is what is required. So over the last many years, we've had multiple collaborations around the world, and really this sort of work requires very large data sets, well-defined phenotypes, and this is often the sort of the Achilles heel, because the phenotypic definition of coronary artery disease varies in different centers, and someone who's had an acute MI is quite different than someone who's had 3-vessel disease and bypass surgery at an early age. So we're talking about a burden of atherosclerosis versus a sudden event that may be more a thrombotic event than definitely due to widespread atherosclerosis. But we have in our more recent data 60,000 cases from and about 123,000 controls from 48 GWAS studies. So this group cardiogram now with C4D. In this sort of collaboration, because the data is not as good from all sites as others, averaging about 9 million SNPs after imputation. And this is a paper that we published in 2015. Majid Nekpe, who's in the audience, is the first author in this, and actually did a lot of the 1000G imputation for it. So this is simply a Manhattan plot. You're not supposed to read anything that's there, but we require a p-value of 10 to the minus 8 for GWAS significance, because there are about, if you look at the haplotypes we're an estimating about 1 million markers across the genome, so Bonferroni correction requires a p-value of 10 to the minus 8. But here the y-axis is truncated at 10 to the minus 20. So a number of variants, and this is really kind of the map to date. 2007, we discovered 9p21 only because it was a low-hanging fruit. It was obviously there. And as the years have gone by, more and more, most recently with the 1000G meta-analysis and UK Biobank data, another 114. So what we have, 160 replicated loci across the genome with GWAS significance. So this is really a complex disease. You know, you consider the number of variants that have a significant effect, albeit a smaller effect. And if you go to a more conservative p-value, that is an FDR-Q less than 5%, there are actually about 304 replicated loci. And this sort of just gives you the idea. So the majority of these SNPs are relatively common, so they're not super rare, and the allele frequency may be as high as 49% as it is for 9p21, or as low as 2%. But the important point is that the effect size of each, the hazard ratio, is low, from about 1.03 to 1.09. The important exceptions are 9p21, where for early coronary disease, the hazard ratio is about 1.3, a little bit less for garden variety later onset coronary disease. The other important loci in terms of a high effect size are lipoprotein A, which we know is a categorical risk factor, and the PCSK9 locus. So, first point, the majority of these loci are not associated with known risk factors. So this is kind of the new information here, because these are the risk factors that we know about, and these are the ones that are used in sort of a Framingham equation or other risk engine. So plasma lipids, blood pressure, diabetes, and obesity, still important, but the new knowledge really goes beyond that. So this is just a schematic, and in terms of those 160 GWAS significant loci, they cover a number of important processes. So first of all, LDL is there, and of course, if it wasn't there, we'd be very concerned about the methodology. Lipoprotein A is there, really a strong causative risk factor. Triglyceride-rich lipoproteins, and this is kind of important because when I was a medical resident many years ago, 30 years ago, triglycerides were kind of not considered a risk factor for coronary disease. I mean, the dogma was, well, triglycerides are associated with risk only because they're associated with obesity, low HDL, et cetera, et cetera. But clearly we know from the GWAS studies that triglyceride-rich lipoproteins are clearly a causative risk factor for coronary disease. Whether the causation is related to the triglyceride component of these large lipoproteins or the cholesterol component is not really clear. It probably is the cholesterol that is actually atherogenic. And we also see signals for insulin resistance, so related to diabetes. But more importantly, what's coming out is a lot of genes that are related to vascular remodeling, the integrity of the extracellular matrix. And some of these have been particular interest in our lab, and I won't go into the details there in terms of the functional studies, but I think the extracellular matrix has kind of been ignored and clearly very, very important. I mean, we do have these patients with very high levels of LDL and they never develop coronary disease, so there obviously are not only risk but protective factors at the level of the vessel wall that may confer immunity to some of the usual risk factors. Peter's not here today. But inflammation and innate immunity is clearly there. Katie's here. There are inflammation people in the audience. So yeah, so very clear that this is causally related to coronary disease, but also nitric oxide signaling, vascular tone, this of course relates to blood pressure, and thrombosis per se. So the first point to make is that the heritability of coronary disease is about 50%, you know, give or take. So about 50% is due to clinical environmental factors and the rest to heritable factors, and most of these are common variants with a small effect size. But, you know, based on the initial analysis, and this is from Majid's paper in 2015, the identified loci, even though there's 160 of those, only explain about 25% of the CAD risks that's assumed to be heritable. So where is the other 75%? Well, Majid in the audience has done a lot of very nice work, but he should be explaining it, not me. But there are other statistical approaches to determine heritability and to determine causation. So Majid has used these mixed linear models. So this involves, it's because the overall contribution of SNPs to a trait can be determined without testing the SNPs individually, so without having that burden of a GWAS significant p-value. Now, to do this, it really is important to have individual genotype information, which of course we don't have from all of the other groups around the world for a number of reasons. So when Majid did this analysis, he's able to explain about 55% of the predicted heritability of coronary disease. And we have the same sort of analyses more recently, really using all of the information across the whole genome. What's more important in Majid's analysis determined that the genes involved in inflammation relatively are much more important than, for example, genes in the central nervous system or elsewhere. But the important point here is that genes involved in inflammation account for about 1/5 of SNP-based heritability. So clearly this is important. And of course, we have had some success with anti-inflammatory therapies. Unfortunately, the ones that work best are very expensive and probably will never be mainstream, but it is something that, you know, remains a target, a potential target of therapy, and certainly lots of investigation. Well, of course, this is a complex disease, so it's not simply related to single SNPs. Even single SNPs affect various processes in terms of including gene expression, etc. But what's happening now is this striving to go beyond the single SNP. These are the sorts of analysis Majid Nikpay is doing. I'll talk a little bit about what he's done recently. But so the relationship of genetic variation to disease clearly is not a linear process. It's interactive, it's complex. There are a number of different levels involved, from genetics to genomics, gene expression, to effects on proteins, to effects on metabolic parameters. So really there's this need to integrate multiple layers of information. You can only do that with, you know, really sophisticated computing. Majid does all of his work with Compute Canada, so way beyond what we have here in terms of IT. And of course, there's a number of ways you can integrate these data, from obviously expression QTL analysis, something we've been doing for some time, but also looking at the relationship to mirrors, which can affect many processes, proteins, etc. And we also have to remember that there is tissue and cell specificity. So getting back to the idea, if you have a genetic variant that affects expression of a gene, it sort of leads to the credence for the hypothesis that that gene may be causative. The trouble with expression QTL analysis is it's often very cell-specific. So we have data sets out there like GTEx, and they have pretty good data on different tissues. They don't often have good data on single cells. And so, you know, there's a lot of information there that, in spite of what's out there, that is still missing. So one of the first approaches we used a few years ago was pathway analysis. So this is kind of the entry into this sort of network analysis. So obviously it makes sense that perturbations in complex networks and in cellular pathways are really likely to be involved in the initiation and progression of coronary disease, same for a number of other complex phenotypes. So the point here is that weakly associated variants, who by no means meet a p-value of 10 to the minus 8 or even an FDR of less than 5%, they can provide important information regarding biology if they cluster within a pathway. So if you've got a bunch of genetic variants that don't individually meet significance, you're finding that they're all within this pathway, then it becomes very likely that that pathway is important. So I won't go into the details here for this interest of time, but as in all of these sorts of studies, we did a study where we had discovery population, 7 studies with about 10,000 cases and controls in each, and then replication in 15,000 cases, 55,000 controls. So it's really doing meta-analysis and then replicating. And so when we chose the pathways that had a p-value of less than 0.1 or an FDR less than 5%. So this is different than the single gene. We come up with a whole pile of pathways that look very interesting. So I'm just going to summarize it here. So this is a little bit different than what I showed you earlier in terms of the single genes and where they were in terms of different processes. The pathways that really come out as being important, lipoprotein transport, of course, is there, but really want to make the point, extracellular matrix integrity, clearly very, very relevant to coronary disease. TGF-beta signaling, vascular smooth muscle cell proliferation, and there's major interest in this area. We know that within the vessel wall, in humans, probably the majority of lipid-loaded foam cells are actually of smooth muscle cell origin rather than of monocyte macrophage origin. And once again, innate immunity. So you can kind of look at this and say, well, you know, these are four pathways that clearly are important, and they go well beyond what we knew in the past. This is another analysis that Majid has done recently, published in Cardiovascular Research just last year. It's, you know, we've kind of focused on a locus and does it affect expression of an adjacent gene, and that's expression QTL analysis. But these loci that are significant can affect expression of a microRNA. And that microRNA, in turn, can have a multitude of effects, not necessarily local effects, but other effects across the genome, so trans QTL effects. So what Majid did was to use a data set which we fortunately had microRNA-seq data for about 710 individuals. We'd like to have more, but this is an expensive thing to do. The problem is there's not a lot of microRNA-seq data out there. Hopefully we will have more shortly, but we had to do replication in two studies, the GWAS study and the Framingham study. And what Majid did was to identify a GWAS significant microRNA QTL for 143 of these circulating QTLs. So this is just the Manhattan plot, so it's for mirrors, not for anything else. In green are those that were replicated in GWAS, where we didn't have data from Framingham. In brown, those we replicated in Framingham. There's no data in GWAS. And for a few of these, the data was available in Framingham and GWAS. So, you know, once again, we're really held up by the fact that there isn't as much microRNA-seq data out there as yet. And so just to summarize a few of these are microRNAs that are causally associated with a cardiometabolic trait, and after Majid did two-sample Mendelian randomization analysis. So we come out 19085P associating with LDL, A1C, fasting glucose, 199 with LAD, with LDL cholesterol, and MIR can be with coronary artery disease. And that's just, you know, a few of the many. So I'm just going to show you one of these because it's kind of interesting. It tells you how we get from these kind of analysis to what we actually do in the lab. So this slide is complex. It's not really complex. So we have these two SNPs here, and 174561 in complete LD with this other SNP. So these two SNPs are basically tightly linked, so they're almost the same SNP, although one may be functional, one isn't. And so these, what's interesting is that this locus and this SNP is within the fatty acid desaturases 1 gene. It has been associated with lower levels of LDL cholesterol, and it's been reported to have trans QTL effects on the LDL receptor. That is, it's not adjacent. This is acting across the genome. So what Majid was able to show with SMR analysis is that this SNP exerts its effect on LDL and these glycemic traits in part by altering the expression of MIR 19085P. So this is really important. So it is an important MIR, seemingly, because this is how this effect in LDL is actually exerted. And so what we've done there, and this is done by Caitlin Beeler in my lab, who's just finishing her master's and who refuses to stay for a PhD, which is too bad, because she has a really exciting project. This is really interesting. So the minor allele of this SNP is associated with higher levels of this MER, higher circulating levels of this microRNA, and lower levels of LDL cholesterol and lower levels of fasting blood glucose and hemoglobin A1C. And so first of all, she was able to confirm in Q87 cells that the presence of the G allele results in higher levels of the miR-1908 and about a twofold increase relative to the A allele. So this is really interesting. So if we can somehow increase this MER, this may be helpful in lowering LDL. She then went on to show that this is a microRNA in the liver that actually responds to cholesterol. So cholesterol loading results in increased levels of the MER, and cholesterol depletion results in a decreased level. So very interesting, because we didn't know anything about the regulation of this MER. But more importantly, she has shown that although we were not able to modify the levels of the LDL receptor at the plasma membrane by overexpression or knocking out that MER, with mimic treatment, so you really increase the level of the miR-1908 5P in the liver, what we're actually finding is that, you probably can't see this clearly, but we're seeing an increased ratio of the mature LDL receptor versus the cleaved LDL receptor in the plasma membrane. So that is important, because the uncleaved LDL receptor is basically active in binding LDL. So this may be the mechanism by which this MER, this SNP that affects the expression of this MER, is associated with lower levels of LDL cholesterol. So there's lots of work to do here. We have to do pulse chase studies in a hepatocyte model to determine if we can actually define this at the level of pulse chase. So that's a lot of lab work to come. Okay. So having taken you through all that, I'm going to try to give you a few clinical pearls, I hope. So I think all of this information really has been useful, and really in a number of different ways. So first of all, precision medicine. Pharmacogenomics, I'm not going to talk about that today, but of course, Heart Institute, Derek So's project, I mean, this really, being able to do bedside genotyping of genetic variants that have a significant effect on the metabolism of clopidogrel, so you know when to use clopidogrel rather than the more expensive medications, or warfarin. That can be really very useful, and I think we've learned a lot there, and there's probably more to learn. We've also learned about endophenotypes. So there are actually genetic variants that affect insulin secretion versus insulin sensitivity. That may be of some guidance in terms of which drug you're going to use in the clinic, whether it's something that drives insulin secretion from the beta cell versus insulin sensitivity in the periphery. Also, target drug discovery, and I think this is really exciting. I'll tell you about it. I think we're going to see more of repurposing of existing agents. So when we find that there is a genetic variant that explains the sort of unexpected effect of a given drug that's in use for something else, that may well be of clinical importance. So in terms of endophenotypes, this is, I kind of made the point initially that, so for these CAD collaborations with 48 groups around the world, you know, some have defined patients as having had an MI or some just as CAD, but we do have information for a lot of these individuals and whether or not they've had angiographic coronary disease but haven't had an MI, and those that have had MI risk, but no risk of, no seemingly burden of atherosclerosis. So this is just one example. The ADAMTS7 locus associates with the risk for having atherosclerosis, but not MI, whereas the ABO locus, which actually has a lot of pleiotropic effects, it associates with the risk of MI, but not with angiographic CAD. So those patients who basically don't have a burden of atherosclerosis but have a sudden MI. Obviously, there's many other loci that affect thrombosis, but this clearly is one of them. Secondly, target drug discovery. So you all know about PCSK9, really in large part a Canadian discovery, finding these individuals who were deficient in PCSK9 and actually protected from coronary disease. But the GWAS studies have shown a very strong association of alleles at the PCSK9 locus and risk of CAD. But there's a number of other important drug targets that are up and coming. And I think a good example is the ANGPTL3 locus. So this is angiopoietin-like 3. And first of all, it was noted in populations, and a lot of this was done in the Amish individuals in northeastern United States, but other groups as well. Individuals who had a loss of function variant, so this is a coding variant in ANGPTL3, definitely were protected against coronary disease, also had lower levels of plasma lipids. So this discovery, which, you know, we're talking about, this is 2017, very, very recent, has led to the development of an agent. This is a bit like, you know, the PCSK9 inhibitor. So a monoclonal antibody versus ANGPTL3, it's called evinacumab, and it probably will be mainstream within a couple of years. Really interesting. So it lowers triglycerides by 75%. And we have few other remedies that lower triglycerides, also lowers total cholesterol, and in animal models of atherosclerosis definitely protects. So I think the important point to make here, because we've had a lot of failures in drug development, for example, the CETP inhibitors, very little signal from the genetic point of view that they would be beneficial, is really minimal. LP-PLA2, really no signal that they'd be a benefit, and nonetheless, those were taken forward and billions of dollars spent in them. If you have genetic evidence for a link between a genetic variant or something affecting expression of a gene and outcome, it markedly increases the likelihood that a new agent is going to be successful. So, you know, the advice to big pharma is that, you know, pay attention to these data, and I think they are now, because deciding to go ahead with evinacumab was certainly a very good idea. So this gets us into Mendelian randomization, which is really a super simple concept. Unfortunately, there are a number of confounders, which I won't go into today. But for example, if you have a biomarker linked to a disease, and I think a good example is C-reactive protein. So CRP, definitely associated with disease. LP-PLA2 is definitely associated with the risk of coronary disease. But is it causative? Is it kind of somehow a bystander that is affected by something else? Are those biomarkers likely to be reliable drug targets? If the genetic variants, the single nucleotide polymorphisms that affect the expression or the level of that biomarker also associate with disease, then it certainly gives credence to the hypothesis that indeed this biomarker is likely to be causative. And once again, we do have caveats there. So, for example, LDL cholesterol, that's certainly true. All the variants near these various genes that affect LDL strongly associated with CAD. True for lipoprotein A as well, and of course LPA, we have a new antisense oligo in the works that will lower this biomarker, this risk factor, very effectively, whereas we haven't done other things in the past. Once again, IL-6, you know, the canakinumab story. It's not true, however, for CRP or LP-PLA2. So these should not be drug targets, and of course the big intervention studies, the big two studies against LP-PLA2 really failed miserably. The other concept that we derive from this is important in terms of how we look at prevention. So at the moment, you know, the trouble is most people are started on cholesterol when they've had cholesterol-lowering therapy, a statin, when they've had their event or if their levels are high, or they're not started in any treatment until the age of 40 or 50. What the genetic studies tell us is that if you have a lifetime of genetically lower levels of LDL cholesterol, so this is from birth, there's a much greater impact in terms of the reduction in ultimate risk of coronary disease than if you're simply on a statin for 5 to 7 years in these studies. So obviously treating someone with a statin for 5 years, I mean, there's benefit. Everybody talks about residual risk. Well, the residual risk is basically because the medication wasn't started early enough, as far as I'm concerned. But it really gives credence to this idea that we should be thinking about preventive therapy a lot earlier. So life years of high LDL is really like pack years of smoking, which of course we think of all the time in clinical medicine. The other sort of interesting concept, and this hasn't really been developed, but I'm just going to give you one little vignette here. Can we repurpose existing therapies? This is work done by Brent Mitchell at McGill University, a colleague of mine, and a really bright fellow that we should have recruited here but didn't. So it gets into this idea of vitamin D. So, you know, I think everybody over the age of 50 is on vitamin D to prevent the risk of osteoporosis, and, you know, does it work or it doesn't work? So what Brent did, he determined the SNPs near these bunch of genes that affect plasma levels of vitamin D. And really what he found was that there was no association with fracture risk. So again, there isn't really a great deal of data that vitamin D has any really beneficial effects on fractures. But what he did find is that these variants markedly affect the risk of multiple sclerosis. So it kind of fits in with the idea that MS is more common in northern climates where there's less sun, so obviously less vitamin D synthesis in the skin. But in part because of this, you know, vitamin D, it's not that you're going to use it to treat MS, but in populations that are at risk, it really puts a lot of emphasis on treating with vitamin D earlier in life to reduce the risk of ultimately developing this not common but nonetheless debilitating disease. Okay, the last thing I'm going to talk about is predicting risk. So this is kind of something I'm particularly interested in, and it'd be nice if we could get it off the ground a little bit more. So polygenic risk score. So it gets back to the idea, first of all, traditional risk factors, moderately predictive individual risk. So important, but that's not the whole story. But we have lots of preventive therapies, so we can find out who's really at risk. You know, we start them on a statin at the age of 20 rather than the age of 50, or something else that is going to be protective. So it's a very simple concept. So a polygenic risk score is simply you take the number of risk SNPs and you adjust for the effect size. So if you have something like 9p21 where the effect size is large, it'll end up getting a larger score. And so there are various ways that this has been done over the years. First of all, only using the SNPs that are genome-wide significant, or more recently, including genome-wide SNPs with no p-value cutoff, so what's called a meta-genetic risk score or meta-polygenic risk score, and this is, you know, with over a million SNPs being used to calculate risk. So first of all, when Sonny Dandona was here, he was a cardiology fellow back in 2010, we simply looked at 9p21 because it does have a large effect size. And what Sonny found, he took individuals who'd had coronary angiography and counted the number of risk alleles so that these individuals have been genotyped. And those individuals who had basically clean arteries, the allele frequency was, you know, close to 40%. Oh, sorry. Those that, gotta go back. Those that had single vessel disease, you know, about 50%, the same as most of the population. But when you get up to 3-vessel disease, the allele frequency of the risk SNP was much, much higher. So we're much more likely to have 2 copies of the risk allele. So it really shows how even a single variant can have an effect on risk. So more recently, other groups, this is work from Amit Kira and St. Kathiresan, published in 2016. They simply did a polygenic risk score with 50 of the GWAS significant SNPs, so not all of them. And really what they were able to show in 3 separate cohorts, so the atherosclerosis risk in communities, women's genomic heart study, and the Malmo diet heart cohort, individuals in the top third of a polygenic risk score, just based on that number of SNPs, versus those in the bottom third, about a 2-fold increase in risk. So, you know, that's really pretty significant for something that is actually fairly easily obtained. Oops. So if you instead create a polygenic risk score that's based on many SNPs, and this was also Mitkatir and Kathiris published in Nature Genetics about a year and a half ago. They created a polygenic risk score, so you're taking in all of the genetic information across the genome. This is really significant because the individuals in the top 1% of a polygenic risk score actually had a 5-fold increase in risk. So that is obviously much more than any categorical risk factor, and clearly could be very important information. So, of course, you know, if Salim Youssef was here, he would say, oh, well, you know, genetics is one thing, but, you know, it's all lifestyle. It is true that if you have a high genetic risk, it's why you probably should know if you have a high genetic risk. But, for example, this was work from Hindi and colleagues. They were able to show that there was a statistical interaction between a genetic risk score and a simple risk factor such as smoking. So individuals with a high genetic risk, if they never smoked, obviously much lower risk than those who've been smokers or are currently smoking. But we see this for the mid-risk and even the low-risk individuals. So, you know, this is perhaps there is some hope that giving individuals genetic information will have an effect on behavior. Amit Kher was also able to show, going back to these 3 studies where they had the polygenic risk score based on 50 SNPs, that for each of these 3 prospective studies, that individuals with a favorable lifestyle, those are in shown here, if they had a low genetic risk, risk was even lower, but those in a high genetic risk category, the top third, if they had a healthy life, definitely that was associated with attenuated risk. This wasn't really a statistical interaction study, but, you know, kind of makes the point. So obviously important. So are we there yet? You know, I think this is quite feasible. So we have this gene titan sitting in the Ruddy genetics lab that hasn't been used because we need some money to get this off the ground. But basically what you can do with an array, and this is the PMR array, but there's various arrays you can get there. So you can have about 800,000 genome-wide markers. This array, you can load it with clinically relevant variants. You can have, you know, coding variants that have a high effect size that clearly are important for, you know, a number of diseases. We're not just talking about coronary artery disease, but, you know, any actionable genetic risk. You can put pharmacogenetic variants on there, so people will know, you know, a priori, you know, how they metabolize warfarin or clopidogrel or statins or something else. But you can also customize it with de novo markers. So you have this option of adding things that are of research interest to an individual or a group. So the point is that this is not expensive. These arrays, you know, the sort of the real cost is about $50. If you add in running the assay and then having someone like Majid doing the bioinformatic analysis, it's less than $100. I mean, what do we do here that costs $100? I mean, like, you know, you can't breathe without spending $100 when you see a patient in the clinic. So I think that is really where perhaps we should be going. And I'm just going to close here with I just came across this on Friday. So Rory Collins. So Rory Collins is the principal investigator of UK Biobank. So I haven't talked about UK Biobank because this is giving us the information that we're already using in terms of our analyses, but very important. So the UK Biobank, they have, in spite of the country that has all its problems with Brexit and whatever else, basically 500,000 people are being screened between the ages of 40 and 69. They intend to follow these 500,000 individuals for 30 years. They have all sorts of phenotypic information, or behavioral information, et cetera, et cetera. They already have 50,000 exomes, so they've sequenced, where the whole exome has been sequenced, so you're picking up all of the, you know, high impact variants. And a large number have been run on basically a gene chip, but they're actually ultimately going to have whole genome sequencing in all of these individuals. Apparently 4 pharma companies have promised to pay for this. Regeneron is going to make exome sequencing available on 300,000 of these individuals as of this year. So that's a huge number of exomes, which is really important. So Regeneron is really being very helpful in terms of this. So when they asked Rory Collins, your opinion, what is the most exciting thing happening in genomics right now? So he didn't say whole genome sequencing. He said the opportunity to implement polygenic risk scores in healthcare systems is something that could have a really big impact on the way in which we provide public health, potentially cost neutral in that it could enhance the efficiency of screening, target those at high risk earlier and low risk later, and by using preventive therapies more efficiently. So he says, what's exciting is that this is a deliverable from genetics that would have an enormous impact on population health. So a lot of people are skeptical about the whole thing, genetic testing, putting everybody's DNA in a chip, but I think it's really the way to go. Okay, I won't get into this. You know what you've learned, probably if you followed it even. I'd just like to thank the people in my lab. So basically, Sebastian runs my lab because I don't. Majid, who's done all of this bioinformatic analysis. Steph is my secretary. Paulina Lau, who's been with me for many years. Anne Tu, who's just finishing her PhD. And Caitlin, who has just finished her master's. So I actually have a very small lab, but I also have to thank all the people around the world who, these collaborations have required a great deal of effort by many individuals. Okay, happy to answer any questions. Thank you.